The aim of this procedure is to measure cytotoxicity of target cancer cells in human whole blood with single-cell resolution. First, a single-cell array of target cancer cells is generated. Human whole blood is added to the cell array, and drugs, such as antibodies, are applied. The cells are then incubated with a cell viability dye, such as propidium iodide. And high-throughput imaging is used to measure the number of dead target cancer cells stained red, ultimately providing drug cytotoxicity efficacy.
The main advantage of this technology over existing methods such as LDH and chromium assay is that cancer cell killing can be done in human whole blood with imaging capability.
This method can evaluate antibody cytotoxicity in vitro before animal study.
Begin by obtaining cell attachment 96-well plate kits or single-cell array 8-well chamber slides. Here, the 96-well plate will be used. Apply 1.2 microliters of the activator to the tube containing DNA reagent from the kit. Cap the vial, invert it, and gently shake to mix the solution. Allow the solution to react for 20 minutes at room temperature. In the meantime, prepare the target cells. In this demonstration, Raji lymphoma cells will serve as the target cells. Maintain them in supplemented RPMI 1640 culture medium at 37 degrees Celsius in a CO2 incubator. To prepare the target Raji cells for the experiment, transfer 5 to 10 million of them to a 15